Where do your traits like eye and hair color come from? Traits are encoded by your DNA, but how does a sequence of DNA get interpreted to make your eyes brown or blue, for instance? It's proteins that give you your traits, and therefore if you have brown eyes, this means that you produce brown eye color pigment protein. DNA is copied into RNA, which is then used to make a protein that corresponds to a trait in your body. All three types of RNA discussed in Part A of this lecture are produced by DNA transcription. Most genes are transcribed into messenger RNA, which will be used to make a protein. However, some genes are also transcribed into ribosomal RNA or transfer RNA. There are three major parts to transcription that we are going to go through: initiation, which refers to the beginning; elongation, which refers to the lengthening of an RNA strand; and termination, which refers to the end of the process. Transcription begins when RNA polymerase enzyme binds to the promoter region next to the beginning of the gene to begin to unwind the DNA. During elongation, the polymerase travels down the DNA strands, continuing to unwind the DNA double helix as it travels. Once the double helix is unwound and the two strands are pulled apart, RNA polymerase is able to use DNA to synthesize an RNA strand. Because DNA is double-stranded and RNA is single-stranded, only one of the strands of DNA is needed to make the RNA. This is referred to as the template strand. Therefore, RNA polymerase synthesizes an RNA strand with bases complementary to the DNA bases in the template strand. What this means is that adenine pairs with uracil, because remember, RNA does not have thymine, it has uracil instead, and guanine always pairs with cytosine. During elongation, RNA polymerase travels along the DNA molecule and unwinds it. The enzyme uses the template strand to synthesize an RNA strand with nucleotides that have bases that are complementary to the bases in the DNA nucleotides. Transcription ends when RNA polymerase reaches what's called the terminator sequence, which is right after the end of the gene, which signals that the end of the gene has been reached. The single-stranded RNA molecule is now complete. mRNA remains linear, but it's actually modified before exiting the nucleus to be used to make a protein. You don't need to know the details of these modifications, however. Both rRNA and tRNA are folded into three-dimensional shapes according to complementary base pairing within the molecule itself. In eukaryotes, DNA is transcribed into RNA in the nucleus. Here is shown mRNA transcription yielding a pre-mRNA molecule, which is then modified into the final mature mRNA molecule. This mature mRNA transcript exits the nucleus and goes to ribosomes in the cytoplasm, where it is used to translate a protein. We know that many genes in DNA are copied into messenger RNA, which are used to make a protein. But how does the language of DNA and RNA, which is nucleotides or bases, get translated into the language of a protein, which are amino acids? This has to do with the genetic code, which translates the sequence of bases and nucleic acids into the sequence of amino acids in proteins. 3 nucleotide bases in the messenger RNA code for 1 amino acid in a protein. The 3 nucleotide bases are referred to as a codon. So this is a messenger RNA triplet encoding 1 amino acid. It's called a codon because it carries the code for an amino acid. The genetic code translates each nucleotide triplet or codon in messenger RNA into an amino acid. We'll learn how to use this table over the next couple of slides. There are actually 64 possible combinations for 3 bases, but only 20 amino acids. So this means that most amino acids are coded for by more than 1 codon. If you look at the table below, I have boxed in the amino acid leucine. LEU. You can see that leucine is encoded for by 6 different codons. This is actually important from a survival standpoint. What this means is that if a codon changes, it does not always change the amino acid in the resulting protein, which could potentially change the function of the protein, therefore harming the overall organism. Where does the sequence of a protein begin and end in the DNA and therefore the RNA? What you have to realize is that there is sequence at the beginning and end of the RNA that does not correspond to amino acid sequence in a protein. Therefore, the ribosomes scan the messenger RNA until they find a start codon. This is a codon where a protein sequence begins and it corresponds to AUG. AUG leads to the methionine amino acid being added to the protein first. Let's use this start codon to make sure we understand how to read the table below. The first letter is A, so find A on the left-hand side of the table. You now know that you are looking for a codon in the third row. The second letter is U, so find the U at the top of the table. You now know that you are looking for a letter in the first column. So now we've narrowed it down to the first column and the third row. From that point on, it's easy to find the third letter, which is G. You see that AUG corresponds with methionine. There's also 3 different stop codons that correspond to where a protein sequence ends. These are UAG, UAA, and UGA. Notice in the table below that it simply says stop because these stop codons do not code for an amino acid. You should use these stop codons to practice using the table further. There are 3 components needed to build a protein. The first is messenger RNA, which contains the information from a DNA gene that determines the amino acid sequence of a protein. The second is tRNA. tRNA carries amino acids to be added to the protein being synthesized. Finally, ribosomes. Ribosomes consist of rRNA and proteins, and ribosomes form a platform to which the mRNA and tRNA bind and on which the protein gets built. All 3 types of RNA are needed to build a protein. Recall that mRNA is a linear strand of nucleotides that carries the message or code for the protein. rRNA combines with proteins to form ribosomes, and tRNA is a 3-dimensional structure with a binding site for an amino acid on one side and the anticodon on the other side. Messenger RNA is used to build a protein during the process of translation. Just like transcription, there are 3 parts to translation. Initiation, which refers to the beginning, elongation, which refers to the lengthening of the protein being built, and termination, which refers to the end. Translation begins when the start codon of messenger RNA, which is AUG, binds to the complementary anticodon of tRNA carrying the methionine amino acid to begin protein synthesis. The small and large ribosomal subunits also assemble around the messenger RNA. Elongation begins when a second tRNA with an anticodon complementary to the second mRNA codon binds, and the ribosome adds the second amino acid to the protein. This process repeats until the protein is complete. In other words, a third tRNA with an anticodon complementary to the third mRNA codon binds, and the ribosome adds the third amino acid to the protein, and so on and so forth. Translation ends when the stop codon in messenger RNA signals to the ribosome to terminate protein synthesis, and both ribosomal subunits disassemble. Translation begins when the tRNA anticodon recognizes the AUG start codon on messenger RNA. The large ribosomal subunit joins the small subunit, and a second tRNA with an anticodon complementary to the second codon in mRNA binds. Then a third tRNA with an anticodon complementary to the third codon in mRNA binds. As the ribosome slides along the mRNA, the polypeptide chain is formed. Each tRNA is carrying a different amino acid that is determined by the anticodon sequence. This process continues until the stop codon in mRNA is reached. During translation, amino acids are strung together in a straight line on the ribosome and must be folded into three-dimensional shapes in order to be functional. Recall from Unit 2 that there are 4 levels to protein folding: primary, secondary, tertiary, and quaternary structure. Enzymes catalyze formation of chemical bonds, and specialized proteins called chaperone proteins stabilize partially folded proteins in order to allow them to fold into their final shape. Many disorders actually result from improperly folded proteins, including cystic fibrosis, sickle cell anemia, Alzheimer's disease, and mad cow disease. A linear amino acid chain is folded into its final three-dimensional shape with the help of chaperone proteins.